let's get started and go way back to when you decided to become a scientist. What motivated you to pursue this path?
I think I grew up curious about the natural world. So where I grew up, actually in Wisconsin, where
Oh,
you are in
really?
school, I lived in the Arboretum, which you may know,
Here in
and
Madison?
here in Madison.
Oh,
Yeah.
that's super cool.
And so my life was all about going out and catching snakes and turtles. And at one point, I had a giant snapper turtle that was laying eggs, and I was taking care of the babies. I had pet raccoons and other things. And so I was always just really observing nature and enjoying that. And at some point, I thought, that's what I want to study as an undergraduate. Eventually, that led to this idea that maybe I want to be a marine biologist, but growing up in Madison, Wisconsin,
I
far
think I
from.
had that phase too.
And that phase was very real for a lot of us back then. So I graduated from high school in 1983. And then at that time on Sunday nights, you could watch this television show starring Jacques Cousteau, who was an inventor of scuba diving and had these specials on Sunday nights where he would go explore the underwater world. And it was, really exciting to listen to this French guy talk about now we'll descend into the protein depths in search of the elusive shark who is going to be sleeping.
Love
And,
the accent.
and that just loved it. Yeah. And so I actually did my undergraduate work at a marine lab
Okay.
and got exposed to doing research there by an amazing pair of scientists who ended up hiring me after I finished undergrad to work with them for a year as a hired scuba diver.
So
And
you were actually a marine biologist for a little bit.
I was a professional scuba diver who dreamed of becoming a marine biologist. And I learned to study some things about the nervous system at that time. So I was studying things about chemosensation and vision. And I ended up going to graduate school actually to get a PhD in neuroscience with the idea that that would be my angle on how to become a marine biologist. Because I figured, it's hard to become a marine biologist, but maybe if I have a neuroscience degree, that'll help. And I was very naive. People would ask me questions back then. I remember interviewing for graduate school, and somebody asked me, like, "So you're interested in learning more about vision? Are you more interested in the retina or the visual cortex?" And I said, "What's the retina?" Well,
Oh
if
wow, yeah.
needless to say, I didn't get into that graduate program. And so I did end up studying vision in a marine organism, in a horseshoe crab.
Okay.
And it was, built on the research that my PhD mentor had done and others before him. And my scientific grandfather, a guy named H. Kepler Hartline, actually won the Nobel Prize for medicine or physiology for his discoveries on how contrast enhancement works in the eye of the horseshoe crab. And so I felt like, this is real science and I'm really excited about it. Um, and when I finished my graduate work, I had become interested in how the horseshoe crabs could see equally well day and night.
Hmm.
And so I got into daily rhythms at that point, and I was really interested in how does that work. So then I went and did my postdoc training. And that training was what really trained me to be a circadian biologist and really, I think, trained me to be the kind of scientist that I am today. So I'm a blend between an engineer and a biologist as a result of my mentors and what they taught me. And when I was a postdoc, it was at a time when NIH funding was moving away from funding things like horseshoe crab research and more towards model organisms where there was genetics and molecular tools. And so all of a sudden, my dream of becoming a marine biologist became a realistic dead end. And I switched to studying mammals. And now I live in St. Louis. You can't get farther from an ocean. And so I'm a neuroscientist with a dream of going scuba diving. Well, the first thing you'll notice is that the lab got a lot bigger. When I started, it was one person, and
Just you?
just me. Um, and so, one thing that really shapes what your lab is like is the people who are in it. And so, over the years, I've had 12 graduate students and 12 postdoctoral fellows. It's always been a fairly small business. But what we study is very much a reflection of people's interests in the lab.
Mhm.
And when they come to the lab, we work together to really think about what are we going to do in the time that you're here as a trainee. And so over the years, I've been surprised to see my lab gradually transition from doing a lot of very basic biology, understanding how circadian rhythms, how we wake and sleep works in normal health in a normal system, to more and more we're asking about how can we fix this system when it's broken in diseases like glioblastoma and preterm birth, where my lab is now involved in 2 clinical trials.
Super cool. Can you tell us a little bit about what they entail?
Sure. So the glioblastoma project is the most mature of the 2. There, in collaboration with my colleague Josh Rubin, who's a pediatric oncologist here at Washington University, we were both fascinated that molecules that he was targeting for therapy were molecules that my lab was studying as varying with time of day. And so
Okay.
we started to ask the question, do tumors have daily rhythms? It turns out glioblastoma, the most deadly form of brain cancer, has beautiful daily rhythms that synchronize to the host. And so we've taken samples from patients and showing that isolates of glioblastoma have circadian rhythms in gene expression, sensitivity to chemotherapeutics, and other things in a dish. And then we started doing what's called a retrospective clinical trial, where we've looked at data that had already been collected in the hospital, just asking, what are the outcomes for people who were treated in the morning with the standard of care versus people who were treated in the evening. We discovered that there was about a 6-month extension of life for those who associated with those who were treated in the morning.
Oh, wow.
And there was a massive effect because the drug was made standard of care because it extended life by about 2 to 2.5 months. And here we're saying that it actually extends life by 6 months if you take it in the morning compared to in the evening. And so that's part of this story. The clinical trials that are ongoing now are prospective trials testing whether we can actually improve outcomes by considering the daily rhythms of the patient.
What projects are ongoing in your lab that you're excited about?
Sure. And, if the listeners are high school, undergraduate, or graduate students, these are the kinds of projects that we love to give to trainees. And so I mentioned that my lab studies the basics of how this system works. And so some of the projects are really designed to help us understand what are the signals that these cells use to synchronize to each other so that we have coordinated daily rhythms in our body. We've discovered different neuropeptides that are released by neurons. We've also discovered signals that are being controlled by glia, the astrocytes in the brain. We're very interested in how these different signals are integrated to generate a coherent orchestra of rhythmic players. We're also interested in how that system controls different aspects of physiology. So every morning before you wake up, you have a surge of cortisol, and we don't know how that happens. We also don't know how it is that females have higher cort than males, and the daily rhythm in females depends upon ovulation. All of these things are under the control of the circadian clock, and we'd like to be able to trace that pathway from the clock to its outputs to understand how is it that we have sex differences in hormones that are secreted at particular times of day. So those are some ongoing projects in the lab. Maybe one of my favorite bizarre projects that we have going on in the lab is we collaborate a lot with engineers who
Okay.
are inspired by synchrony. And when you think about it, synchrony is everywhere in our lives. Just planning traffic patterns to get to work is an exercise in understanding synchrony. There will be a lot of cars during rush hour. We need to figure out patterns to get everybody to work, and then there's sort of a lull, and then there's another rush when everybody heads home. And
Mm-hmm.
so synchronization is a phenomenon that's everywhere in our environment, including daily synchronization. So things like power usage. Even we have times of day when everybody turns on their air conditioner in the summer, and times of day when everybody turns on their heater in the winter.
Mm-hmm.
Those things are circadian by virtue of the fact that we are circadian. And so engineers like talking to me about, the principles that we learn in understanding how cells synchronize. So my lab's developed methods to map out the connectivity between cells, between neurons, for example.
Okay.
And once we understand the connections, the topology of those circuits, we want to understand, is that telling us something about optimal system design? Since evolution has converged on 20,000 pacemaking neurons in your brain, coordinating everything about your day and night. And so we have become very fascinated with sort of rules of topology, of connectivity in the brain. And then the last things that are ongoing are those projects that relate to human health. And so we have, there's an area of cancer that you may have learned about called cancer neuroscience, or neuroscience of cancer, where certain cancers actually seem to be growing faster as a result of neural activity. And so my lab is interested in, well, you have daily rhythms in neural activity. Does that mean you have daily rhythms in signaling to tumors causing them to grow? Meaning maybe we should be trying to block that signal to slow tumor growth. And that could be true not just in the brain, but in all sorts of tissues. That's a fun question. So my PhD mentor was a man who did not mentor a lot of graduate students. He had just a handful of graduate students during his career. Bob Barlow was absolutely obsessed with beautiful writing. So from him, I really learned, hopefully, something about how to express my ideas. He was very passionate about science communication. He went to area schools, and he would be sort of a visual illusion magician and explain to them how their visual system worked. And that's something that I've inherited from him, this responsibility to talk to society about what we're doing. So I lead the St. Louis Neuroscience Outreach initiatives. And from my postdoctoral mentor, a man named Gene Block, who was really, I credit him with how I think as a scientist today. He really taught me about gain of function and loss of function experiments, how to test for necessity and sufficiency. And he taught me how to think about the best experiment. He taught me, don't do the experiment until we've decided it's the best experiment. And for those listeners who don't know about the last lecture, you should all go watch the last lecture. It should be available to you on YouTube. It's also a book. It's the story of a professor who at the time was at Carnegie Mellon University and had been diagnosed with terminal cancer. And so he titled this lecture The Last Lecture because he anticipated that he wouldn't have much longer to give a lecture about why do we do what we do as researchers. And in the lecture, he mentions one person by name. He mentions my postdoctoral mentor, Gene Block. And he mentions him in the most positive way in the context of a lesson, which is basically when you're faced with a brick wall, like administrators who tell you can't do that, or a mentor who tells you that experiment is impossible. You go around it and you find your Gene Block. You find somebody who will facilitate you in your dream. And so for me, Gene Block really was that person and continues to be that person. He's now the chancellor of UCLA, and I can write to him anytime I have a question. He'll be right back to me.
How would you describe your personal mentoring style?
I would describe it as adapted to the mentee. So I do try to talk less, listen more. I try to understand where your weaknesses are and build your weaknesses to strengths. I try to understand where your strengths are, and I don't have to worry about those. I just have to remind you they're strong already. So I try to be supportive, and where I need to help you get over your limitations, I try to make it clear that this is just part of making them stronger and better. I've been humbled many times to realize, for example, neuroscience today doesn't study blank. And so some of those lessons have been things like, right now in the news, there's all this discussion about transitioning in your gender identity. And many of those folks who are going through that transition, there is no understanding of the neurochemistry of what's happening in the brain. There's very little known about the consequences of the therapies that are being prescribed. And yet we have politicians making decisions about who can do what, physicians and patients. And I think that it was the Endure program that helped me appreciate that this was a big gap. And they've taught me about gaps in our understanding of the consequences of solitary confinement, short-term and long-term. They've helped me appreciate, these students are just great at teaching me about, cultural differences and how they can really bring new perspectives to projects. I'm a big fan. Diversity makes the project strong. Feels great. We're still pretty young in the WashU Endure program. We are in our 8th year as a program, so we've just had our first undergraduate now get their PhD.
Really cool.
And that was wonderful to see. But it's been really fun to watch as people are growing up in science and can tell their own stories and can give some credit to the Endure program and say that it was really, it was a big step up for them, big boost.
Definitely.
So as scientists, we have to pick and choose our battles. And some of those battles are, in the lab, overcoming the challenges of this experiment is not working. And that is probably always going to be my priority first to the things we're doing in the lab right now. When we write grants, convincing our colleagues that these are really important ideas to be tested, that's an important part of my job. When we write papers and we discover things, it's really important for me to write those stories in a way that they're of broad interest and they're understandable. And so that's an important challenge. But I think you're also asking about a bigger society than that, a bigger community than that. And so, I raised 2 boys with my wife, and that got me into their classrooms very early on. I've been really lucky. I've had some really cool research experiences that have taken me all over the world. We talked about my scuba diving. I was a scuba diver in Antarctica. You tell that to a kid and to that's what I want. And so I feel it's really important to share the excitement of what I do. Every day is about discovering something that people didn't know before. And when we've discovered something that may be helpful, I feel obligated to spread the gospel. And so I do find myself more and more talking to representatives in Congress. I go to Congress and I talk to them. And as I mentioned, I'm a member of several scientific societies, and I'm involved in their government affairs committees to ensure that, we're advocating for best practices based upon the scientific discoveries.